Rouge, Louisiana, and he will be speaking on in vitro diagnostics for stroke enabled through microfluidics and single molecule detection. Okay, Steve.
Okay, so while we're waiting for this to come up, I'm going to thank Richard for the opportunity to come here and speak today. And let me point out something. I specifically targeted a disease, stroke. But frankly, the platform that I'm going to be talking about today is good for any scenario where expression profiling needs to be done. So first of all, I want to thank Claudia for the nice introduction. I'll be able to breeze through some of these concepts, but I'm going to add a little bit of our slant on this. So let's take a look at stroke for just a minute. This is an interesting disease because as of right now, to the best of our knowledge, there's no in vitro diagnostic for stroke. Why? And the reason for that is simply because of the blood-brain barrier. A lot of markers that are spilled in circulation or blood don't make it across the blood-brain barrier. So a lot of times people have been using, for example, a cardiac panel to translate that over to stroke with not too much success in terms of the clinical sensitivity and specificity for one simple reason, again, the blood-brain barrier. So the problem, again, the diagnostic techniques that are typically used for stroke is this: CT or MRI. You cannot do that in the field. That has to be done at an emergency room, for example. So you can see this is a third leading killer in the U.S. And really what you would like to be able to do is take the diagnostic phases of this disease into the field. And I'll share with you why that is indeed the case in just a minute. All right, now the other thing that you have to remember is that there's two different types of stroke: ischemic versus hemorrhagic. Ischemic is actually when you have either an embolism or a blockage in the artery in the brain, or hemorrhagic is when you've actually ruptured one of the capillary walls and you have major leakage of blood into the brain. Now, this is the problem. MRI and CT cannot differentiate between those two types of stroke. And that's a big challenge because one of the common modalities to treat at least ischemic stroke is tissue plasminogen activator. It's a blood thinner. It'll help release that clot. However, you would not want to treat a patient with hemorrhagic stroke with this particular therapeutic. Now here's the other problem too, is that we need to provide information on a very short time scale. We have about a 3-hour window to administer this particular drug. So if we have to transport a patient to the hospital, undergo a CT scan, in many cases it's too late to administer that therapeutic. And indeed, less than 2% of the total population of those patient populations that are undergoing ischemic stroke are not administered effectively using that therapeutic. So this is a big analytical challenge, because think about what we need to do. We need to make the measurement preferably in the field, and we need to do a rather complex measurement. Now, I haven't even told you what type of measurement we need to do, and that's the next thing that I need to talk about. Okay, so here's the central hypothesis. We're going to do a gene expression profile on a certain subpopulation of blood cells to diagnose stroke. And that's still not the whole story. We need to differentiate between ischemic and hemorrhagic stroke too. Critical to do that. So we're going to do that, and again, look what we're going to try to do here. Gene expression profile. We're going to look at our collaborators are looking at which genes that we can use and those that are being dysregulated as a result of these stroke events. Our challenge is to develop an assay in the hardware to do this expression profiling. Now, this is different than what you heard in the previous talk. In the previous talk, we only need really quantitative data. Yes or no? Is there a mutation or something indicative of that particular bacterial species? Here, we need quantitative data. We need to understand which genes are being upregulated or downregulated and how are they differing from their basal level. So it's a little bit more challenging in that respect. Now, if you look at stroke, there are certain subpopulations of cells in whole blood that are responding preferentially to this particular event. And most of these cells or the markers that we're going to be looking at are being recruited as a result of tissue damage in that brain. And some are similar to those that you can use for the cardiac scenario. Some are different and unique, and you'll see some of those shortly here. All right, so blood. It's probably it's one of the dirtiest analytical samples you can ever envision. And this is why. Look at this, and I'm not going to read through all this, but look at all these different types of cells that are in whole blood. Now this is the challenge that we're confronted with too. Only certain subpopulations of the white blood cells are responding to this stroke event. So let's think about this for a minute. And you can see here, neutrophils are not checked off here. So we have, for example, these CD4 positive T cells, and they're responding to this stroke event. And if we want to take whole blood and try to expression profile one gene that might be expressed across this entire population, the leukocyte population, our results are going to be skewed. Because remember, the expression is an average of all the cells that have some expression level of that gene. So if neutrophils has a basal expression level of a particular gene, it's going to wash out because of their large contribution to the total leukocyte population by, for example, the CD8 positive T cells. So we need to pull out these specific subpopulations and look at those specifically. That's critical in order to make the results effective. All right, so our collaborators at Downstate Medical have done an expression profile, done a tremendous amount of work on trying to figure out which genes are responding to this tissue damage in the brain, and also which cell populations are expressing those differences preferentially. And there's a list of them here. I'm going to just focus in on amphiphysin. It's involved in endocytosis, and it's predominantly expressed in T cells. Remember, there's multiple subpopulations of T cells, right? The CD4 positive and the CD8 positive. All right, so when they did this work at Downstate, the trauma center, brain center, stroke center, here's the assay that they followed. They cleared out the red blood cells, lysis cells, solid phase extraction, messenger RNA, so on and so forth. All right, so now, not only is the infrastructure high in doing those assays, but the processing time is about 11 hours. Of course, obviously, once you've selected, this is a discovery-based approach. Obviously, once you've selected the gene panel, you can go back and use RT-qPCR as well. So we're kind of thinking about a new paradigm to actually do expression profiling. And the one thing you'll see in these boxes right here, there's one word or one abbreviation that's not there. PCR. We're getting rid of it. And why? Why are we doing that? You know what? It amazes me. PCR is not a very good quantitative tool. Have you ever looked at the yields that are generated as a function of cycle number or copy number? It's very non-quantitative. You have to do a lot of processing in order to make it, from a statistical perspective, make it quantitative. So you probably, at least from a quantitative perspective, you'd really like to be able to negate the need of doing PCR. That's the analytical perspective on that. The other thing, too, is that Claudia pointed out that actually doing PCR on a microchip can reduce that processing time. No doubt about that. However, again, there is a bottleneck in terms of how fast you can do a PCR reaction, especially if you want to do a multiplex PCR reaction. And it's typically limited by the kinetics of the enzyme. So anytime you can negate the need for PCR, it makes it very attractive in terms of a quantitative analysis. So what we're doing is, the front end of this is actually quite similar to what you saw for Claudia. Cell lysis, doing a solid phase extraction, reverse transcription. In this case, we need to convert the messenger RNAs, the transcripts, into cDNAs that we can do the analysis on. And then we're doing what's called a ligase detection reaction, and I'll show you that on the back end of my talk. The other thing that we're doing, too, is we're doing digital counting instead of analog counting. Everyone who does optical spectroscopy knows that photon counting is much better in terms of quantification than analog measurements. So, okay, that's Jim Weidenford, and wrote a series of papers on that about 10, 15, 20 years ago. So we're doing digital counting. We're actually counting the copies of the material that we're generating to get this quantitative data, and I'll show you some results of using that strategy. All right. This is nothing new to everyone. It's the electronic microprocessor. It's basically the hardware of your computer. What's really nice about computers? Well, you can choose the RAM, you can choose all the other peripherals in it in order to accommodate your needs. So it would be very interesting if we could do that with microfluidics as well. Oh yeah, just so you don't forget. This is actually an IBM, this is one of the IBM computers that was, I think this was published in about the early 60s. You can see how far computers have come. Probably this is where we are with analytical instrumentation, right? We're still at this stage. We still have a long ways to go to make this a little bit more portable. It's footprint size of a PC. We're getting there though. All right, so we are working on developing fluidic microprocessors. Now, and Claudia brought this up in her talk too, but there's several reasons for doing this, and I'm going to kind of give a little bit different slant on this. So what we do is we have a fluidic motherboard, and we plug task-specific modules directly into it. The nice thing about this architecture is we can personalize our experiment to our needs. So we can generate a toolbox of modules that can plug into these universal motherboards and to accommodate a particular application need. So in our particular case, we need to select out certain subpopulations of the white blood cell population. We need to lyse those cells that we selected out. We need to do a solid phase purification of that material, and in this case it's messenger RNA, not genomic DNA. Do a reverse transcription in this LDR and single pair FRET. So the nice thing about this architecture is that you can pick, and Claudia alluded to this briefly, you can pick the material you need to suit the application to optimize performance. And let's take a little look at this. So for this cell selection, there's a couple things that we need there, right? We're going to use a positive selection, so we're going to attach antibodies onto these discrete modules. So we need some surface that we can functionalize. The other thing that we need is we need something that is moldable, something that we can shape into the particular structures that we so desire. The next thing is thermal lysis, and a lot of these thermal reactors are poised on this motherboard. So for this motherboard, in order to carry out those thermal reactions, we are going to need something that has a fairly high glass transition temperatures. So we're using polymers here. Let me back up for just a minute. We're using exclusively thermoplastics for this. So we need something with a high TG to accommodate those thermal reactions. A lot of these thermal reactions we're doing, I'll go pound through this pretty quickly, is using these continuous flow processes. Isolate the RNA using a solid phase extraction. We need something that's going to have a high propensity to allow that material to condense onto it selectively. And then the reverse transcription, again, is done on this motherboard. And then finally, the readout, this thermal reaction and the readout. We're going to use fluorescence. In fact, actually, we're going to use single molecule fluorescence. So we darn well better have a material that has a very low autofluorescence yield. Aha, but don't forget, it must be moldable or machinable as well. All right, so we use polymers, and I've been actively watching Chip Shop and their work over the years. They've done a tremendous job with commercializing a lot of these polymer-based chips. Why do you like to use thermoplastics? And let me just go to the next slide. And it's kind of an easy answer because the infrastructure to produce those chips is already there. CDs and DVDs. These are well-established technologies to make structures. All right? But that's not the whole story with microfluidics. You're not just making channels or structures. You're adding reagents to them to affect some type of biological process. So you need to have a material that's biocompatible, that you can modify its surface, and also, again, as I pointed out, it's machinable using conventional infrastructure, for example, injection molding or hot embossing. Either one of them is fine. And there's some other little physiochemical properties that you need to be aware of. The optical properties, the electrical properties. Are you doing electrophoresis with that particular chip? So here's some materials. These are common ones. This is, and Claudia showed this. This is cycloolefin copolymer. It's a copolymer. That's really interesting because copolymers, you can change the monomer composition of that copolymer to change its physiochemical properties. Refractive index, TG, you can fine-tune all that. Polymethylmethacrylate is particularly elegant because it does have a fairly low glass transition temperature, but it shows very good optical properties. And I like this plot here because it's kind of giving you a rundown. There's a lot of different materials on here, and I won't go through that. But there's several things that we can learn. Well, if you're just looking at the intrinsic fluorescence that these materials are generating when you're exposing them to either blue, red, or deep red light, you can see immediately that some materials behave better than others. And also, you probably don't want to use your conventional argon ion laser if you're using any type of thermoplastic with optical fluorescence detection, because you're going to get a large background level. It's going to exceed that, typically what you see with glass. So you have to be a little bit careful in that respect. All right, so we have to attach things to this polymer surface. What we're trying to do, let me back up one more point and say one more point too. When you make these chips for diagnostic applications, and I said this morning, typically you're using it for, you're not doing this for research, you're using it for one sample and you're discarding it. So you'd like to keep the chip cost low. You'd like to keep it on the same price range as tighter plates. And you know what? Frankly, we've done an extensive cost analysis on this. It's not really the material that you're using and the machining or the molding that's expensive. It's the reagents that you're putting in the darn chip. Antibodies, your finishing, assembling, all those can add up to over 80% of the total chip cost in many cases. So you have to be careful of that. So we'd like to generate strategies that allow the molding to make some of the passive and active elements in the chip. So we don't have to add things to it, like beads, anything else you can think of, membranes or whatever. So one of the things that you can do with polymers, and this is actually ubiquitous across a number of different materials, you can expose them to UV light, which generates a photooxidation reaction. And you can generate a plethora of different type of scaffolds from which you can elaborate from. So for example, one of the functional groups you generate is carboxylic acids. When you expose this to UV light, you can plasma treat it too. It does the same thing. But once you've done this, then this is an anchoring point for any type of an amine-bearing compound, primary amine-bearing compound that you can think of. So you can actually covalently strap antibodies directly to these functionalized surfaces that have been exposed to UV light as well. Yeah, one more little caution. Some of these materials have very high water contact angles. They're hydrophobic. All right, it makes them a little bit difficult to pump liquids through them, or you generate bubbles during the liquid transport. So you can change that also by exposing it to UV light. It has a dramatic impact on the contact angle. It makes it more wettable with aqueous materials. All right, so here's one of the modules. This module here is used to select out these subpopulations from the total leukocyte population. In fact, actually, we can do this directly out of whole blood. We don't need to deplete the sample of the red blood cell population. Usually those are they're about 3 orders of magnitude in higher numbers than the white blood cells. So what we're doing here is we've actually decorated these channel walls. This is the selection bed right here. We've decorated these channel walls with CD4 or monocytes, and in fact I didn't write that down, I'm sorry about that, that's CD14. So we can actually simultaneously input one blood sample and pull out the monocytes and the T cells that are CD4 positive. And there's no reason that we can't put the CD8 antibodies here and the CD4s to look at different subtypes of those T cells. And we could also look at we could look at B cells here as well. So in one pass-through, we can actually pull out the 4 subpopulations that we're particularly interested in looking at. How is this done? How do you get these antibodies into here, 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 and here? Well, actually, there are several ways to do that, the way this is designed. One way to do that is to expose only this section through a UV mask to activate or generate that functional scaffold. And when you influx those antibodies, either any one, Only that area that was exposed to the UV light through a contact mask will be used for immobilizing those particular antibodies. You could do that sequentially then. Let's see how I'm doing here. Okay. All right, so this is just a work in progress. One of the things I want to point out here is how many cells, and I know someone's going to ask me this, what is the sensitivity of this measurement? So let me just hit it off at the pass here. The relevant question is how many cells do you need to do the measurement? And that's predicated on the copy number of the messenger RNA in the cells and the input volume of the blood that you feed into the device. So with a 20 microliter input, and we assume that we're measuring this now, we assume we only get a 10% collection efficiency, then 500 T cells and monocytes per microliter are required to make that measurement. And I'll have some interesting things to say about that at the back end. All right, now, this is a very interesting phenomena here. This is a material-dependent process. Whenever you lyse cells, chemically or electrically or thermally, really doesn't make a difference, you generate a lot of junk. And you typically, before you do the molecular analysis, you employ a solid phase extraction, as Claudia alluded to. Well, what we can do is you can take polycarbonate and expose that to UV light. You can take a solution of nucleic acids and suspend it in an immobilization buffer. And that immobilization buffer is polyethylene glycol and sodium chloride. Those nucleic acids have a tendency to condense onto that photoactivated surface. And that's shown in the next panel here. So the nice thing about this, again, polyethylene glycol and sodium chloride with a little bit of ethanol in there too. The nice thing about this is you can change the composition of PEG and sodium chloride to selectively pick out a different sized nucleic acid population. So when we use for the messenger RNAs, we're using about 6% polyethylene glycol and about 0.5 molar sodium chloride. We're free of any genomic DNA, mammalian genomic DNA. We only get the mRNA population. In fact, actually, we're getting the total RNA. I need to be a little bit more specific in that respect. So you can see, even when we're operating this under fairly high shear conditions, it's not that terribly high, but we're still getting very good integrity in terms of the composition of those RNAs. This is the total RNA population. We're looking at the ratio of the 23S to 16S, and the ideal ratio here is 2, and we're about 1.8, so it's not so bad. Now, what is the recovery? Actually, the recovery on this is about 75%. So we're recovering about 75% of the total RNA that we're putting into this device directly from a whole blood sample or a whole cell lysate. And it's simple to do, right? All you do is expose this material to UV light and you're ready to go. The other thing that we've done in order to increase the bed capacity, we use this molding step to not only make these fluidic networks, but also these pillars. So actually the load of this is pretty much predicated on the size and the spacing of these pillars. And we have great flexibility in how we can choose that. And then Claudia showed you this too. This is this continuous flow PCR. And actually it's PCR, but remember, we're not doing PCR. There is one thermal cycling reaction we're doing. It's this ligase detection reaction, and I'll show you that on the back end. But one little interesting thing we're doing with this is that we're making a dual depth channel. Now, why would you want to do that? Okay, that just shows you one of the nice attributes of molding. You can make multi-level structures. You can make structures on the front and back side of the substrate. You can make slanted structures. You can do a variety of different things with the molding. The reason we do that is that this is the bottleneck right here. I don't care what enzymatic reaction you're doing. The bottleneck is how fast the enzyme works. It's kinetics. So what we do, without increasing the footprint too dramatically, what we do is we increase the cross-sectional area of this channel. So when this fluid comes in, it operates at a fixed linear velocity or volume flow rate. When it gets to this section, it slows down, and this is exactly where you want it to slow down because that's the bottleneck, right? It's the kinetics of the enzyme. And you can do these reactions in under, we've done 20 PCR cycles in under 20 minutes and got a fairly good yield, 1 minute and 20 seconds, and got very good yields on this. So I'm going through this kind of fast because you heard a lot about that in the previous talk. So let me get to this back end now. This is the last module. Remember, The solid phase extraction module, the cell selection module, and all the thermal reactions are done on this motherboard. This module is the readout module. And basically what this does, we are doing single molecule detection. So we're reading fluorescence out online on the output end of this thermal reaction that we're doing. And again, I'll show you the assay in just a minute. Now here's another little problem with this. You have to remember, you're never going to make a viable diagnostic from looking at one marker. There's some examples where that you can get away with just looking at one marker. But we're going to have to probably look at a panel of markers. In fact, actually our colleagues tell us we're probably going to have to look at 5 or 6 different markers. So we need to do multiplexed analysis. So basically what we do is we do spatial multiplexing in this case. It minimizes some of the criteria on the hardware end of things. So we have an array of channels that each one corresponds to a different reaction that we're using in order to read out that result. All right, so spatial multiplexing, similar to an array, really. So this is very interesting here, because now we're making some active components, optical components, using molding. So we have an embedded waveguide here with a coupling prism, and then that serves as the foundation for this fluidic network. That's roughly about 0.5 microns in terms of depth and about 200 microns wide. Now, why are we doing that? Why is this height so important? Well, basically what we're doing, remember, whenever you launch light into a waveguide, a planar waveguide, you got light leakage due to this evanescent properties. So this light leaks into this adjoining solution layer due to this evanescent property, so this material of the waveguide. So if you launch this in close to the critical angle, you get a very deep penetration into that intervening solution or adjoining solution layer. The other thing that we've actually got going on here is we got some coupling prisms here, these relay optics that are machined on the top of this, the bottom face of this, or the bottom face of this fluidic network, and you can see that right here. So we published this several years ago, but here's how you can make embedded waveguides. We're using polymethyl methacrylate as the substrate with a channel that's been embossed into it, and then we connect onto that, or just conformally seal on that, a piece of PDMS that serves as a stencil. Then we add a COC melt into that, and this waveguide and the coupling prism is made with cyclic olefin copolymer. Those two materials have a different refractive index. So because of that, this waveguiding will be, the light propagation will be pretty much confined to this waveguide. And this just shows you an example of that. Well, of course, the intensity or the amount of light leakage in that intervening solution depends intimately on the launch angle. And you typically want to do this at very close to the launch or the critical angle of PMMA and COC in this particular example. Yeah, you can also make, this is another thing you can do with molding. Typically you want to have good replication fidelity during the molding, so you make sure the polymer fills up this cavity in the molding tool. But you can use what's called a hot intrusion process. You only get partial filling, and you can actually make curved structures or lenses. You can actually, depending upon the molecular weight of the polymer and its surface tension forces and the temperature and pressure you use, you can actually change the radius of curvature of that. All right, so now the last thing that I wanted to mention here, and I'll have to do this a little bit quickly, is on the back end, reading fluorescence or single molecule fluorescence out on the back end. So remember what we have here. We have this ligase detection reaction, and we have this thermal reactor that's continuously generating products. Those are being fed into the detector online. So we're not doing a batch measurement, we're doing an online measurement. And again, the one thing that I want to take home to you is two things. We're doing digital counting, molecular counting, and we're eliminating the need for processing steps because of the sensitivity that's developed through the use of single molecule detection. All right, so not just the chip is important, but the peripherals around that too. And one of the cumbersome things with doing fluorescent single molecule detection is the extensive amount of equipment that's required to do that. And this is when I was at Los Alamos National Laboratory, this is the instrument that we put together in order to do single molecule detection. Not very transportable into the clinic, I agree, especially in the field. So we reduced the footprint of that, and here's the new one that we have. Actually, we just published a paper on this. I didn't mention on here, it's in the Analyst, just appeared in January this year. And we've actually reduced this footprint down to a 5 by 7 foot PCB board with all the support electronics to make the measurements and the optomechanics as well. And all this is all fiber coupled. So there's a diode laser on here as well as a single photon avalanche diode. All right, now the assay. This is really important too, because what we need to do is to recognize certain mRNA transcripts that have been pulled out of this sample. And the way we do that is using a reporter sequence within that particular transcript. So we have 2 oligonucleotide primers, this is the cDNA, that recognize that reporter sequence. If there's complementarity between these primers and this cDNA, they're ligated together. And appended on the back end is a stem structure and a donor and an acceptor. So when we ligate those 2 primers together, it forms a molecular beacon, brings these 2 dyes, the donor and acceptor, into close proximity. In the absence of ligation, or the transcript is not there, there's no ligation. So there's very little, if any, energy transfer between these donor and acceptor molecules. And there's the dyes that we use. Remember, we're using polymers. We have to push out into the red region of the electromagnetic spectrum to mitigate any issues with autofluorescence. So we're looking at this amphiphysin gene, cDNA from this amphiphysin gene. Did a 20-cycle thermal reaction, a ligase detection reaction, 2 temperatures, 65 for the ligation, 95 for the denaturation. And you can see these little spikes that are generated when that amphiphysin gene is fed into this, and the cDNA that's generated from this messenger RNA from amphiphysin. Each one of these peaks corresponds to a single beacon or a FRET process. And in the absence of that cDNA, you get a pretty clean background. What makes this really nice is, again, the name of this expression business is getting quantitative data. You can see the dynamic range here is actually, it's much larger than what I'm showing up here to 10,000 copies. But we have great linearity with correlation coefficients down on the low copy end. And when we do this using conventional real-time qPCR, these are the results we get. Actually, not too bad. Here's the correlation coefficient, but we have a little bit of problem in the low copy number regime. It's a little bit difficult to differentiate the expression differences on this low copy number end. So what I basically showed you today is that microfluidics coupled to single molecule detection is very elegant. It can provide you a number of things: automation, reduced processing time, and high sensitivity, and very good levels of quantification. Single pair FRET for doing genotyping, or actually you can do genotyping of circulating tumor cells or any low copy number material that you so envision. And you can do this for messenger RNAs, as I pointed out today, stroke for real-time reporting. So finally, I want to thank the funders of this particular group, the project, the National Institutes of Health, National Science Foundation, my colleagues, especially Allison Baird. She just moved from NINDS to Downstate Medical. And finally, the people who did all of the work that I talked about today, Jason, Brandon, and Paul and Jerry have been intimately involved in some of the single molecule work. And then a lot of my fabricators, Matt and Maggie Vitek. So with that, thank you for your attention. I'll be happy to answer any questions that you might have. Thank you.
Thank you, Steve. This